Good morning and welcome everyone to another Athena Talk and Hellenic Mediterranean University Sci Cafe Talk. It's a great pleasure, an honor actually for us to have today Professor Nektarios Tavernarakis from the University of Crete and the ISL 4th, IMBB 4th, excuse me. A short introduction about our speaker. He's one of the most distinguished uh, academics and professors in Crete and in Greece in general. A few words about him. Currently, he is a professor of molecular system biology at medical school of the University of Crete, research director at IMBB 4th, and president of European Molecular Biology Conference sector. Also, he is the head of neurogenetics and aging laboratory at IMBB. Previously, he has, held several distinguished positions, such as vice president of the European Research Council, president of board of directors at Fourth, and president of governing body at European Institute of Innovation and Technology. Additionally, he holds several professional memberships, such as European Academy of Science of Arts and American Association for Advanced Science in Europe and European Academy of Sciences. He got his PhD from the University of Crete, and also he has postdoctoral experience from the United States at the University of Rutgers in New Jersey. His research focus is on molecular mechanisms, cellular metabolism, sensory transduction, and development of genetic tools for biomedical research. So his topic, the discussion of today's presentation is on the role of cellular turnover and detoxification mechanisms. So we are very glad and happy to have you today with us, Professor Tavernarakis, and we are expecting a very fascinating presentation. Thanks a lot.
Thank you very much, George. Kalimera, καλό μήνα σε όλους. Thank you very much for having me. It's an honor for me to be here with you today and present some of the work that we are doing related to neurodegenerative diseases and aging. So I will start with explaining why we're doing this. Why is this important? Why is it important to study neurodegenerative diseases in the context of aging? And I will give you some information that I think makes it very clear why these two phenomena, neurodegenerative diseases and aging, are connected. As you can see here, the human population is aging. So we have a gradual lifespan extension for humans, as you can see, beginning in the 1700s, around the advent of antibiotics that took care of many of the diseases that would eliminate patients very early on. So from then on, we have a very steady, as you can see, increase in human lifespan, which is now approaching about 85 years of age in the Western world. And actually, what makes, I think, this observation even more pronounced, even more striking, is that the segment of the population that is of 85 years of age and older is actually the fastest growing segment of the population right now in countries like in Europe, the United States. These are some measurements, some information coming from Sweden and Japan. As you can see, people from 100 years of age and above, or 105 years of age and above, are actually the fastest growing segment of the population, even more steep than the exponential growth. So this, of course, creates problems. It doesn't come without consequences. And the consequences are that diseases which are associated with aging, that used to be rare, are now becoming very frequent. And among them, I think the most well-known group of diseases is neurodegenerative disorders, such as, for example, Alzheimer's disease. As you can see here, if you compare a normal brain with an Alzheimer's disease patient brain, the differences are obvious. There is pronounced neurodegeneration. The structure of the brain that is responsible for memories, the hippocampus, is completely gone in the patient, which of course makes it impossible to form new memories or recall old memories for that matter. But it's not just neurodegenerative diseases. Other diseases are also associated with aging and are now becoming more and more frequent, and we see this in our societies. For example, cardiovascular diseases, metabolic diseases like type 2 diabetes, cancer, and stroke are now the most frequent pathological causes of death in Western societies. Now, focusing on Alzheimer's disease, since we introduced this particular neurodegenerative disease, what is happening in the brains of these patients is actually the nerve cells, neurons, degenerate because mainly of two reasons. The first is the deposition of beta amyloid extracellularly. This beta amyloid protein is forming plaques, shown here. And intracellularly, we also have the formation of aggregates of another protein called tau. So these two proteins, when they aggregate either extracellularly or intracellularly, essentially deprive these neurons of nutrients and choke them to death. This is what happens. This is how these cells degenerate. Of course, we don't really understand the molecular mechanism responsible for neurodegeneration, but at least we know the triggers, what really is responsible for initiating this cascade of neuronal death. And this is becoming a very big problem for human societies, as I've told you already, but these are some numbers so that you can get a perspective about how big the problem is. So the World Health Organization is actually estimating that in the coming decades, there are going to be more than 100 million, even more than 130 million people affected from some sort of dementia and Alzheimer's disease. By the way, Alzheimer's disease is one type of dementia, the most severe one, we could say. But there are other types of dementia, neurodegenerative diseases that are linked to memory deficits. And all these neurodegenerative diseases are going to account for more than 100 million people worldwide, which essentially becomes a pandemic even bigger than the recent pandemic of the COVID virus. Just to get an idea, get a grasp about the magnitude of the problem. And here we don't have a vaccine or an effective therapy to tackle these diseases. So it is actually very important to really put an effort in addressing this problem. And recently, there have been efforts, and maybe you have heard about some drugs, some compounds that have been developed and are being tested in the United States. Some clinical trials are ongoing in other countries as well. About antibodies, essentially focusing on how to tackle the aggregation of tau and beta amyloid using monoclonal antibodies. So two of these monoclonal antibodies are already in the market and can be used in patients, but the effectiveness is very low. And actually, there are a lot of side effects that are quite severe for some patients. So at this moment, essentially, we don't have an effective treatment of Alzheimer's disease or any other neurodegenerative disease for that matter. And that is the reason why we need to really put more effort into understanding the molecular mechanisms responsible for neurodegeneration and then trying, after we've deciphered these mechanisms, trying to develop effective strategies. So this is the motivation behind the work that we are also doing in the lab. We are essentially trying to understand how nerve cells degenerate. And when it comes to neurons, to our nerve cells, actually, these cells are quite peculiar and quite unique because, as you may know, these are non-renewable cells. Some of the neurons that we have in our bodies were actually born even before we were born, and they will literally die, most of them, after we are pronounced dead. So they will live for the entirety of our lifespan, tens of years, even more than 100 years for some of us if we are lucky. So thinking about a biological system, a cell, in this case a neuron that has to survive for that long, these cells need to be able to detoxify themselves and maintain their homeostasis for very long periods of time. And usually, when talking about biological systems, we are always considering them as being quite fragile, quite easy to kill, quite easy to destroy. It's actually very easy to kill a cell. But these neurons, they can survive for many, many years, and not just survive, but also function and transduce electric signals in our brains. So they are highly differentiated cells. They look like cables. Some of them extend for more than 1 meter. Some of our motor neurons, the nerve cells that control muscles, actually have a length of their axon that is more than 1 meter, starting from our brain, our central nervous system, and going down to specific muscles. They are very differentiated, and they are, of course, dependent on a lot of energy to carry out their function. Neuronal cells are actually notorious for being the most energy consuming cells in our bodies. Although our brain is by mass only a small part of our body, actually it consumes more than 20% of the total energy input, of the total energy that we absorb from our food. So they really consume a lot of energy, even just sitting there idling, not doing anything, not transducing any signals, because they need to maintain a polarized plasma membrane in order to be able to transduce electric signals. So even just sitting there, they consume a lot of energy. And they, of course, depend on mitochondria, which is the energy producing organelle in the cell, to produce all that energy. So they should have a very important vested interest in maintaining the mitochondrial population inside them in a functional state, so that they can get the energy they need. And if mitochondria cannot function in neurons, then we have devastating pathologies. Actually, one of the characteristics, one of the hallmarks of Alzheimer's disease is the accumulation of damaged mitochondria. This is something we see if we dissect post-mortem Alzheimer's disease patients' brains. What we see, one of the most prominent features, apart from the beta amyloid plaques and the tau aggregates, is accumulation of damaged mitochondria. So these neurons really contain a lot of damaged mitochondria. So this is what actually triggered us to study this phenomenon of mitochondrial accumulation. So we wanted to understand, is it causative to the pathology of Alzheimer's disease, or is it just a decoration? Is it just a consequence of the general neurodegenerative process that is going on in these neurons? And what made it even more interesting actually was a link to aging, because as I said in the beginning, most of these neurodegenerative diseases are associated with aging. The number one risk factor for suffering from Alzheimer's disease is old age. So we wanted to understand, why is this happening? What is the reason for this association with aging? Because both in hereditary forms of Alzheimer's disease, but also sporadic forms, all the factors that are responsible for Alzheimer's disease are there during our whole life, but we do get symptoms only at a very old age. So we do get a late onset of these diseases. And we want to understand what is the connection with aging and perhaps use this knowledge to sever the connection, to really dissociate neurodegenerative diseases from aging. So the observation that made it important in that respect was the following. We don't just see accumulation of mitochondria in the context of pathology, in the context of Alzheimer's disease, but we also see accumulation of damaged mitochondria in the context of normal aging without any pathology. And I'm showing you here two examples. One, a mammalian, coming from a mammalian system in rats, in rat ventricular tissue. As you can see, we can count mitochondria by just dissecting tissues and putting them under the electron microscope. If we compare rats of 6 months of age with rats that are 24 months old, we see a very clear accumulation of mitochondria. Most of them are damaged. Actually, we know this. We can label damaged versus functional mitochondria using specific dyes. And this is a quantification that shows how this accumulation is happening during aging in a mammalian system. Now, we can also see this phenomenon in another system, in another organism that is used in the lab as an experimental model, and that's Sinorhabditis elegans. This is an organism that we also use in the lab here at IMBB and the medical school. And actually, even more conveniently, in C. elegans, we can label mitochondria using GFP and monitor this phenomenon during aging in live animals. We don't need to sacrifice them and dissect the tissues to count mitochondria under the electron microscope. Here, we can just send a GFP fluorescent protein to be localized in mitochondria using a mitochondrial localization signal in front of the GFP. And then we can even use a tissue-specific promoter to drive the expression of this reporter in any tissue we are interested in, neurons, epithelia, etc. In this case, it's the intestine of the animal. And if we compare day 1 to day 3, day 5, day 10 is already quite old for these animals that live for about 15 days. There is a gradual accumulation of mitochondria. So this is an interesting phenomenon. We have accumulation of mitochondria in the context of neurodegenerative diseases that are associated with aging, but also in the context of normal aging in two very different organisms. So this is an evolutionarily conserved phenomenon, as we say, which means that it must rely on fundamental mechanisms that are common both in nematodes and mammals, and these are two phyla that are separated by millions of years of evolution. So these mechanisms more likely are also operational in humans. And indeed, this phenomenon of mitochondrial accumulation appears to be a common denominator of many pathologies in humans, not just neurodegenerative diseases, but many other pathologies associated with aging, such as, for example, cardiovascular diseases, rheumatoid arthritis, these are autoimmune diseases, other neurodegenerative diseases such as Parkinson's disease and Huntington's disease. In diabetes, type 2 diabetes mellitus, we also see this accumulation of mitochondria. In obesity, in cancer, in lupus, etc. So there is a common denomination here, and that's the accumulation of damaged mitochondria in all these pathologies. So what then is the mechanism responsible for this accumulation, and how is this affecting the function and the survival of cells, and by extension the tissues and the whole organism? Now, if we consider what mitochondria do, actually, of course, mitochondria are energy producing. They produce the ATP that the cells need, the majority of the ATP that cells need to function, to repair themselves, to maintain their structure, and also to reproduce. But in addition to that, they also produce byproducts of metabolism, and that's the reactive oxygen species that is generated by the oxidative phosphorylation process, the respiratory chain inside the mitochondria. And actually, depending on the cell type, between 0.1% to 2% of the oxygen that we consume is converted to reactive oxygen species. Now, these are very active molecules that can cause damage to other macromolecules, such as proteins, DNA, RNA, lipids, etc., which are essential building blocks and functional components of a cell. And of course, the most proximal targets of these species, reactive oxygen species, are mitochondria themselves, because ROS is produced inside mitochondria, and the most proximal targets are mitochondrial proteins, mitochondrial DNA, etc., which of course results in a process of less efficient mitochondria. So as time goes by, because of this damage that incurs during the function, the normal function of mitochondria, and there's nothing that we can do about it actually. This is a byproduct of normal metabolism. So in order to stop ROS production, we would need to shut down mitochondria. Uh, these ROS will eventually cause damage to mitochondria, resulting in a higher degree of ROS production in these damaged mitochondria, because now mitochondria suffer a phenomenon of aging during their lifespan. They begin as young functional mitochondria producing a lot of ATP and very little ROS, but as they accumulate damage, eventually they end up producing very little ATP and a lot more reactive oxygen species. So we have a positive feedback loop here that results in mitochondria getting damaged during their normal lifespan. And this is something that is physiological. It happens in our cells as we speak. And of course, cells would need to have a way of removing those damaged mitochondria if they want to survive in the long run. Especially neurons would need to do that because particular neurons are very long-lived cells. So we decided to address this mechanism and find out what is actually responsible for mitochondrial homeostasis in an organism in the context of neurodegeneration. So to study this, we used C. elegans, and I'm going to just introduce this system to you because it might be new to most of you as an organism. Most people are familiar with Drosophila, for example, or the yeast or the mouse, but C. elegans not so much. Although C. elegans is very well suited for studying phenomena that relate to neurodegeneration and aging, and that's why I'm going to spend just a few minutes introducing this system to you. So C. elegans is a nematode, it's a worm that has a very short life cycle, 2.5 days from egg to adult, and a very short lifespan after that, so about 2 weeks. So it's highly convenient for aging studies. You don't need to wait for months or years for an organism to die to study how it ages. For example, mice take about 2 years. Drosophila might take quite a few months. So, in comparison to those systems, C. elegans offers a convenient platform to study aging. And one other characteristic is that it reproduces clonally. There is self-fertilization. Most of the animals are hermaphrodites, which allows us to establish, as we call, isogenic populations. This is important because aging is a multifactorial phenomenon. It is affected both by genes and the environment, and we need to be able to isolate the effects of a specific genes from the whatever other mutations might be in the genetic background. If we are to study the contribution of a specific gene or a specific pathway in aging. And this is something we can do in C. elegans that we cannot do in humans, we cannot do in the mouse, we cannot do in Drosophila or any other sexually dimorphic organism. So that's an additional advantage. And of course, it's a very well characterized, it's a thoroughly characterized animal We know how it develops from the zygote, from the fertilized oocyte down to the adult that contains exactly 959 cells. We have even recorded all cellular divisions. This is the genealogical tree of all cells in C. elegans, of all 959 cells. So this is unparalleled characterization. We have this kind of information just for C. elegans, no other living organism. And this, of course, allows us to address aging very specifically. For example, muscle aging, neuron aging, epithelial aging, digestive system aging, etc. Because we know exactly which of those cells are muscles. Actually, when it comes to neurodegeneration and neurons, this is the only animal for which we have characterized the entire nervous system. So out of the 959 cells, just 302 are neurons, and we don't just know which these cells are and where they are in the body, but we know how they are connected to each other. So we have the full circuit of the nervous system, which means that we can really follow behavior from the sensory perception stage to the final muscle action that we get. We can even study propagation of signals inside the nervous system, integration of signals, learning and memory phenomena, etc. And this is, of course, another unique advantage. But this, of course, would tell us about aging in C. elegans in the worm and neurodegeneration in the worm when we actually want to understand neurodegeneration and aging in humans. And you might ask, and you might be quite justified to ask the question, okay, if we study C. elegans, we are going to learn about how C. elegans ages and how C. elegans neurons degenerate, but is this going to be relevant to humans? And I think the answer there is yes, because actually most of the fundamental biological mechanisms that we study in C. elegans are also operational in humans in the exact same manner. So, for example, C. elegans mitochondria are indistinguishable from human mitochondria. And there is a very high degree of similarity between proteins and genes in C. elegans to those in in yeast, for example, in fly, in mouse, or even in humans. So this, of course, I think legitimizes doing research in such simple models, taking advantage of all the features and the knowledge that we have in these models to accelerate our research and then go to human, to more complex organisms. And indeed, because the nematode offers these advantages, that is why the first genes that were actually found to influence aging, the so-called gerontogenes, as they are called now, were identified in C. elegans, and then by homology were also studied in humans. And many of these genes we now know affect processes such as insulin IGF-1 signaling, dietary restriction, mitochondrial function, of course, the reproductive system, metabolic processes, the timing of processes inside cells, the chronological mechanisms, and also responses to stress and epigenetic changes. All these phenomena were first identified in C. elegans and then were found to be evolutionarily conserved in mammals and even in humans. So that is the reason why we chose to utilize this system, C. elegans, to study the phenomenon of mitochondrial accumulation in the context of neurodegeneration and aging. And essentially ask the question of what are the mechanisms in neurons that actually are responsible for detoxifying these cells of damaged mitochondria that will incur during normal aging in these cells. And an early interesting observation that actually triggered us to follow this path was the following, shown here. It was observed by Marta Artal, a highly talented postdoc in the lab some years ago. So what Martha observed is that while in wild-type animals, we have this accumulation of damaged mitochondria during aging, which I showed you some slides ago. As you can see here, this is quantification of this phenomenon, day 3, day 5, day 10. Here we actually can label mitochondria using a mitotracker red. It's a dye that labels mitochondria, and we can use it in C. elegans to label mitochondria in any cell type. That's why the more red uh, an animal is, the more mitochondria it has accumulated. So in wild type animals, we do see this accumulation, but in C. elegans mutants that we know are long lived because we have introduced specific mutations in the genes that I told you before affect aging. For example, DAF2 affects insulin signaling and DAF7 affects TGF beta signaling. These are two interventions, two mutations that results in significantly longer lifespan in C. elegans, almost double the normal lifespan. Now, in these animals that live longer, we actually do not see this accumulation of damaged mitochondria. So this actually now is an interesting correlation at this point. It's not proving anything, but it's an interesting correlation. And the correlation is the following. Whenever we live longer, we don't accumulate mitochondria. So is this the reason we live longer, or is this just a consequence of other processes that allow for longer lifespan? And this is what we wanted to address. In C. elegans, actually, we can address directly such questions. And to do that, we would need a way of influencing the number of mitochondria. So dialing up the number of mitochondria or dialing down, reducing the number of mitochondria in cells. We can actually do this in C. elegans using specific mutations that affect mitochondrial proteins called prohibitins. This is not something I don't want to go into details, into technical details about this, just to use it as a way of changing the number of mitochondria at will in cells and then asking what is the consequence in the process of accumulation of mitochondria. So, in wild type animals, if we do this, if we interfere with these two prohibitins, what we see happening is we exacerbate the accumulation of mitochondria. We make things even worse. Now, animals accumulate even faster and even more their mitochondria in cells. But if we do the exact same intervention, so knock down these two prohibiting genes in long-lived mutants, we further ameliorate this accumulation of mitochondria. We make things even better in terms of accumulation. Now there is even less accumulation of mitochondria. And now we can ask what happens in the lifespan of these animals. What is the consequence in the longevity of these animals? And what we observed is actually whenever we make things worse, so whenever accumulation of mitochondria is accelerated, we shorten the lifespan of animals. But whenever we attenuate the accumulation of mitochondria, we further extend the lifespan of these animals. So this suggests that actually accumulation of mitochondria is detrimental for survival. It shortens lifespan. If we can put the brake on this accumulation of mitochondria, if we can decelerate accumulation of mitochondria, then we can live longer. So, of course, now this begs the question of what is the responsible mechanism that underlies mitochondrial accumulation during aging. And this is what we wanted to understand. Are there specific genes that control this? Are there specific processes and pathways that are responsible for degrading damaged mitochondria and keeping the number of damaged mitochondria low in cells during aging? And of course, if you think about what might be responsible, there are two processes to consider. One is the process that is responsible for generating mitochondria in cells. This is called mitochondrial biogenesis. It's the process responsible for generating fresh mitochondria. And then there is also the process of degradation, the turning over of damaged mitochondria. Under normal conditions in young animals, these two processes are more or less balanced. So on one hand, we have generation of mitochondria. On the other hand, we have degradation of mitochondria. And these two processes are all more or less equal. Otherwise, we would, of course, end up with no mitochondria or a flood of mitochondria. There is room for adjusting depending on physiological demands, energy demands, etc. One process may increase, the other may decrease, etc. But here, during aging and in neurodegenerative diseases, what we observe is really runaway accumulation of mitochondria, uncontrolled accumulation of mitochondria. And we see this, as I said, during normal aging, but also in the context of age-associated pathologies such as neurodegeneration. So we decided to dissect this process genetically and find out how we go from here to here, how we end up with runaway accumulation of mitochondria. Now, in C. elegans, we can do genetics. We can use either a genome-wide random mutagenesis to target specific genes and then ask what are the consequences regarding a specific process that we are studying, or we can go the other way. We can use reverse genetics to use RNAi, RNA interference, to knock down specific genes and ask the question of whether or not they are involved in the process that we are studying. I won't again go into the details of how this screen was done, but let me tell you that out of this screen, we identified 3 specific genes that encode specific proteins involved in a process called mitophagy. These genes are NIX, DCT1 in C. elegans, PINK1, and Parking. And I'm going to explain what these genes are and what mitophagy is. So mitophagy is a cargo-specific type of autophagy. Now, what is autophagy? Autophagy is a catabolic process. It's a self-degrading process that is responsible for recycling components of cells. So we have, for example, different types of autophagy. Macroautophagy is the most well-studied one. Macroautophagy entails the formation of an autophagosome around the cargo that is to be degraded. So this autophagosome is essentially a double membrane vesicle that engulfs the cargo to be degraded and then delivers this cargo to the lysosome. And the lysosome, as you know, is the degrading factory of the cell. That's where degradation, the final degradation takes place. And depending on selectivity, we have different types of macroautophagy. Mitophagy is the type of macroautophagy that is targeting mitochondria specifically. And we have both physiological paradigms of mitophagy and pathological paradigms. Let me give you one physiological example. In our red blood cells, in addition to not having a nucleus, as you know, red blood cells don't have a nucleus, but they also don't have mitochondria. And the reason is that mitochondria are eliminated during the maturation of erythrocytes to red blood cells. And they are eliminated through a process of mitophagy. There are 2 proteins, one on mitochondria, it's called NIX, that's the mitophagy receptor, and another protein on the nascent autophagosome called LC3. Through the interaction of these 2 proteins, mitochondria are engulfed, so the autophagosome forms around the mitochondrion, and then the mitochondrion is delivered to the lysosome for degradation. Now, this happens because we don't want mitochondria in red blood cells. The reason being that mitochondria are consuming oxygen, and red blood cells need to transfer oxygen to tissues. So the last thing we want in these cells is an organelle that would consume the very cargo, the oxygen that these cells need to transport to our tissues. So very wisely, evolution has made it so that mitochondria are eliminated and we don't have mitochondria in red blood cells. Now, there is another paradigm more relevant to what we are discussing, which relates to pathological mitophagy. And by pathological mitophagy, I mean mitophagy actually recycling damaged mitochondria. mitochondria that are not functional. So how is this happening? These mitochondria that are damaged are actually recognized because of certain signals that they emit. ROS, for example, is one such signal, but also calcium is another signal that emanates from damaged mitochondria, leaks out of damaged mitochondria. Now, these signals are activating a specific kinase called PINK1. It's a serine threonine kinase that phosphorylates and activates Parkin. Parkin is an E3 ubiquitin ligase that ubiquitinates, puts specific ubiquitin moieties to mitochondrial outer membrane proteins. And that's how damaged mitochondria, specifically damaged mitochondria, are labeled and recognized by autophagosomes and then recycled. So this all happens in the vicinity of damaged mitochondria. And as I described to you, what we identified in our genetic screen is proteins involved in mitophagy. So we identified the C. elegans homologue of Nix, the C. elegans homologue of Parkin, and Pink. Now, as you may perhaps know, Parkin, the name refers to Parkinson's disease, a neurodegenerative disease that is associated with mutations in this protein. And of course, this suggests that mitophagy is also important in the context of Parkinson's disease. But I will tell you a little bit more about that in a minute. So we characterized biochemically this pathway in C. elegans. We used proteomics to identify the specific residues on DCT1 that become ubiquitinated during the response, during the activation of mitophagy through the pink parking pathway. Specific lysine residue is ubiquitinated, and this triggers mitophagy. Now, mitophagy, as you can infer from what we've discussed already, is a pro-survival process. It eliminates damaged mitochondria, so it contributes to the survival of cells and to the response to stress, more specifically oxidative stress that is induced upon mitochondrial damage. And this is the reason why, if we eliminate mediators of mitophagy, we actually shorten lifespan in long-lived mutants, such as those that I showed you earlier, show less accumulation of mitochondria. For example, the DAF2 insulin signaling mutants or the DAF7 TGF-beta mutants. If in these long-lived animals we knock down mitophagy mediators, we take away part of their longevity, which suggests that one reason these animals live longer is because they activate mitophagy. They have activated mitophagy, and this contributes to a higher degree of detoxification in the cells of these mutants, and that's why these mutants live longer. Now, in wild-type animals, if we knock down mitophagy mediators, we don't see any effect. We don't see lifespan shortening. This might be expected on one hand because under normal conditions in wild-type animals, we don't have induction of mitophagy. Mitophagy is kept at low levels, so eliminating mediators of mitophagy or downregulating mediators of mitophagy might not cause a detrimental effect. However, this creates a paradox, and I'm going to explain why here. What we've observed is that upon downregulation of mitophagy in wild-type animals, we have a number of negative consequences. So we have increased ROS production, leakage of calcium out of mitochondria, a collapse of the inner mitochondrial membrane potential that is responsible for the production of ATP, and as a result, we have less ATP being produced. Now, these are all detrimental effects, suggesting that animals with lower mitophagy experience significant oxidative stress. However, as I showed you, there is no effect on lifespan. And this is strange because any manipulation that we do in a living system, in a living organism, in the majority of the times, actually close to 100% of the times, will have a measurable effect on lifespan. Why? Because lifespan is a very sensitive gauge of whatever is going wrong in a biological system. And this is because lifespan is a phenotype that develops over a long period of time. So we have cumulative effects, suggesting that even the slightest effect in the long run will accumulate into a measurable difference. And this is something we don't see. Now, this indicates that there is a compensatory mechanism. So there is something that compensates for this downregulation of mitophagy in these animals. And one such process that might compensate is the so-called retrograde signaling response. Now, what is retrograde signaling? Whenever cells sense that there's something wrong with their mitochondria, they signal back to the nucleus where most of the genes encoding mitochondrial proteins now reside. Only a small fraction of the mitochondrial proteins are actually encoded by the mitochondrial genome, about 13 peptides. Most of the proteins are encoded by nuclear genes. And here we have a master regulator called PGC1 alpha that coordinates the expression of these genes together with other cofactors of the NRF family of transcription cofactors. Now, in C. elegans, we don't have a PGC1 alpha, but we do have transcription factors that are homologous to the NRF transcription factors in mammals. One such transcription factor is SKIN1. So we hypothesize that maybe the reason why we don't see any adverse effects upon mitophagy downregulation is because the retrograde signaling process kicks in, is engaged through the SKIN1 transcription factor. And indeed, what we've seen is that SKIN1 becomes activated upon oxidative stress that takes place upon mitophagy downregulation. And then this upregulation of SKIN1 also results in the upregulation of the transcription of genes that are responsible for mitochondrial biogenesis. These are all genes encoding mitochondrial proteins, actually some of them of the mitochondrial respiratory chain. But interestingly enough, also the DCT1 gene that is involved in mitophagy, that is the mitophagy receptor in C. elegans, becomes upregulated. So whenever we downregulate mitophagy, we don't just upregulate the expression of genes involved in mitochondrial biogenesis, but cells also mount an upregulation of mitophagy itself in order to restore normal mitophagy levels. And this accounts for the compensation that we see happening upon mitophagy downregulation. And if SKIN1 is not there to mediate this compensatory response, then mitophagy can have significant adverse effects, really detrimental effects on lifespan. So in the absence of SKIN1, downregulating mitophagy now results in a pronounced shortening of lifespan of these animals. This suggests that indeed it's the engagement of retrograde signaling in mitochondrial biogenesis that compensates for the downregulation of mitophagy. Now, going back to neurons for the last part of this presentation, what happens if we downregulate mitophagy is that we get really pronounced accumulation of mitochondria in neurons. Neurons are actually the cells that suffer the most by any aberrations happening in the mitophagy process. Actually, let's take things from the beginning. Let's see what happens if neurons at some point in their life detect oxidative stress. This oxidative stress could be because of intrinsic reasons, for example, ROS produced in the mitochondria of neurons themselves, or extrinsic reasons, for example, toxins or other toxic substances entering neurons. Whenever neurons sense oxidative stress, what they do is actually they initiate a retrograde trafficking response where they move their damaged mitochondria back to the soma, where most of the degradative machinery resides, the lysosomal system and most of the autophagic machinery resides. So any damaged mitochondrion, it doesn't matter if it's away from the cell body at some distant synapse, will be transported back to the soma. And this you can see here, for example, let's begin with this diagram. This shows the distribution of mitochondria across the long axon of a neuron. They are forming this distinct puncta along the axon of the neuron. If we challenge these neurons using paraquat, which is a herbicide that induces oxidative stress, or using mutations that affect the respiratory chain and result in accelerated ROS production in mitochondria, what we see is that now neurons cleanse themselves of this damage of these mitochondria. So we have a lot less mitochondria distributed in axons. We can actually even monitor mitochondria in vivo as they move across the axon. So they are migrating, these damaged mitochondria migrate to the soma, and then mitophagy is stimulated in these neurons that suffer oxidative stress to take down damaged mitochondria. So we have two processes. On one hand, trafficking of mitochondria back to the soma, and on the other hand, activation of mitophagy that takes down damaged mitochondria. And these two things happen whenever neurons sense that they are challenged by oxidative stress. Now, if this is not happening, if for any reason mitophagy is perturbed, then we actually render these neurons vulnerable to neurodegeneration. And this we can monitor, we can assess using specific C. elegans models for human neurodegenerative diseases. So in this case, for example, we have generated a C. elegans Parkinson's disease model by overexpressing human alpha-synuclein. It's a protein that is implicated in the pathogenesis of Parkinson's disease, in specific dopaminergic neurons of C. elegans. And we cause some neurodegeneration. So not all of these neurons survive. Not 100% of these neurons survive upon human alpha-synuclein overexpression. We can actually fine-tune overexpression so that we adjust the level of neurodegeneration. But look what happens if on top of that, if on top of alpha-synuclein overexpression, we now downregulate mitophagy. We cause extensive additional neurodegeneration. So these neurons are much more sensitive now to the same amount of alpha-synuclein here. So we have a lot more neurodegeneration and far less cells surviving. This suggests that mitophagy is protective against insults such as overexpression of alpha-synuclein. And in another model, Alzheimer's disease, where I showed you in the beginning that we have accumulation of tau or beta amyloid plaques, what we see happening upon accumulation of tau or beta amyloid is a very strong inhibition of mitophagy. So these cells now have less capacity to cleanse themselves of damaged mitochondria because of tau aggregation and beta amyloid. So mitophagy is perturbed. And finally, in another paradigm, which in this case is more physiological, I showed you two pathological paradigms related to two neurodegenerative diseases. But the question is, okay, mitophagy protects against neurodegenerative insults, but does it play any role in the normal function of neurons at all? And it appears that it does. And in this case, we studied a paradigm phenomenon of associative learning in C. elegans. This is akin to Pavlovian learning. Now, here, what we do actually, this is a typical example of training. we train animals to like or not like specific chemicals. So these animals natively will chemotax very strongly towards specific chemicals that are predictors of food. For example, sodium chloride or isoamyl alcohol. It's as if this is the firmware of the nervous system of these animals. They are born with this capacity to chemotax very strongly towards isoamyl alcohol because it's a predictor of the presence of bacteria. It's a product of fermentation by bacteria, and these animals in the wild they eat bacteria, so they need to locate niches that are rich in bacteria, and that's how they locate their food by using chemotaxis. So natively they will chemotax. It's an intrinsic behavior towards isoamyl alcohol, and this is shown here. These bars show the percentage of animals that chemotax very strongly towards isoamyl alcohol. Now, we can train these animals to not like isoamyl alcohol if we pair this chemical with the absence of food. So now, instead of chemotaxing, actually having a very strong positive chemotaxis index, these animals will show aversion. So an attractant has become a repellent because of the training. Now these animals think that isoamyl alcohol predicts the absence of food, predicts starvation. So they will run away from isoamyl alcohol. But look what happens if we do the same thing upon downregulation of mitophagy in these animals. What we see is there is some behavioral plasticity, so they like less to chemotax towards isoamyl alcohol, but they never learn to avoid it. So this suggests that mitophagy is also important under normal conditions, not just under conditions of stress or conditions of neurodegenerative insults in neurons, but also for the normal function of neurons. And indeed, we can further elaborate on this by using specific upregulators of mitophagy. So not just downregulated, but upregulated, and see if we get any positive effects. And what we see is actually we can restore learning deficits that come from tau aggregation or beta amyloid plaques in an Alzheimer's disease model, in a dementia model in C. elegans, by upregulating mitophagy using specific chemicals that are known to be inductors of mitophagy, specific inductors of mitophagy. One such chemical is urolithin A. It's a compound, it's a natural compound actually, that was shown some years ago in Johan Orex's lab to be a specific inducer of mitophagy. So by inducing mitophagy, we can actually restore learning and memory in a C. elegans model of Alzheimer's disease, suggesting that indeed mitophagy is also playing an active role in the physiology of these neurons, not just in the pathology. And to conclude and summarize what I've told you, what we identified in C. elegans, and actually then found, I didn't have time to go into this, but we then examined the same pathway in mammalian systems, in the mouse more specifically, is that cells, and specifically neurons, possess a pathway, a response mechanism that balances mitochondrial biogenesis and mitophagy. So upon accumulation of damaged or superfluous mitochondria, what we see happening is oxidative stress that results in the activation of a transcription factor, SKIN1, which is an inducer, is a coordinator of mitochondrial biogenesis. But at the same time, also, this transcription factor, as I showed you, induces the expression of a key mitophagy receptor called DCT1. However, this is not sufficient to induce mitophagy, because if it was, that would be non-specific mitophagy that would eliminate also the fresh mitochondria that are produced. We need another layer that provides the specificity, and this is coming from the pink parking pathway that now is activated in the vicinity, specifically in the vicinity of damaged mitochondria that release ROS and locally activate pink1 and parking, which then ubiquitinates DCT1 and activates mitophagy that takes down this damaged mitochondria. And this pathway also interfaces with the insulin IGF-1 signaling pathway that directly regulates skin-1 and DCT1 through another transcription factor, stress response transcription factor of the FOXO family of transcription factors called DAF16. And more specifically, in neurons, mitophagy is playing a very important pro-survival role because it is responsible for the long-term detoxification of neurons from damaged mitochondria. Mitophagy is the process that specifically eliminates damaged mitochondria in neurons, allowing them to survive for very long periods of time. Mitophagy is actually more upregulated in neurons compared to other cell types, providing this layer of protection. I didn't have time to go into the details about how this is happening. As I told you, we have transport of mitochondria to the soma. This involves calcium signaling, but also a number of signaling events orchestrated by calcium calmodulin kinases and the MPK kinase, which eventually results in the activation of mitophagy, elimination of damaged mitochondria, and blocking neuronal cell death, neurodegeneration. If mitophagy cannot function in neurons, then we have, of course, accumulation of damaged mitochondria, increased calcium release, which then triggers apoptotic and necrotic pathways that result in neurodegeneration. And that is how neurons really protect themselves against damaged mitochondria. If mitophagy is downregulated, these neurons become defenseless and are more prone to neurodegeneration. And this was even confirmed in humans because we collaborated with a group in Norway that examined post-mortem brains of Alzheimer's disease patients. And they found that mitophagy markers are severely downregulated, severely less pronounced in the brains of these patients, implicating mitophagy directly in the pathogenesis of Alzheimer's disease. So I will finish here by thanking all the people in the lab, my friends and colleagues at the medical school of the University of Crete and the Institute of Molecular Biology and Biotechnology at Forth, and more specifically Kostas Palikaras, who did the genetics part of the work that I presented to you today as part of his PhD thesis. Kostas is now a professor at the Medical School of the University of Athens, and Irene Mionaki, a postdoc in the lab who did the biochemical studies and the proteomics analysis I showed you. And thank you very much, of course, for your attention. I hope it was not too complex, too complicated, or too tiring. And I would be happy, of course, to take any questions that you may have.
Thank you very much, Professor Tavernarakis, for the presentation. It was really exciting, and of course, the way that you speak, the enthusiasm is very inspiring for our young researchers. Thank you very much. Now I would like to kick off the Q&A session. So please either write your questions to the chat or raise your virtual hand. And I already see Shmuel Yerusalmi would like to ask a question. So please unmute yourself and direct your question to the speaker.
Yes, thank you very much. You hear me?
Yes, we can.
Yes.
Okay. Thank you very much on a very interesting and insightful presentation. And I want to ask you a question. How, according to your opinion, cellular turnover can support prevention diseases of dementia, Alzheimer, and another diseases of cognition?
So, based on what I've discussed today, I showed you mitophagy is a process that could be targeted to try to increase resistance against neurodegenerative insults and perhaps also use this as a way of prevention for neurodegenerative diseases. As I showed you, mitophagy is neuroprotective. So if we have a way of easily upregulating, if we have a way of activating mitophagy, specifically in neurons, then we can, in principle, defend against neurodegeneration. This is something that we showed in our models, both C. elegans and mammalian cells in mouse neurons. And of course, it remains to be seen if this will be an effective strategy for humans. And I know now that there are such studies going on, trying to develop specific mitophagy inducers. So chemical compounds that are inducing mitophagy specifically without any other adverse effects. I showed you some of these compounds here. Urolithin A is one such compound. Another is NMN, nicotinamide, for example, that are being now studied in the context of the human pathology. But I have no specific information about that. So this is in the very early stage of trials in human cells.
Thank you very much.
Thank you, Shmuel, for the question, and Professor Tavernarakis for the reply. Professor Kavoulakis, yes, please.
Well, first of all, thanks a lot for your presentation. Very interesting stuff. I have a very trivial question. It is well known that there are places around the world where people live way longer than the average, like the island of Ikaria in Greece, for example. Is there any obvious connection with what you presented to us, you think?
Yes, indeed. There are these blue zones, as they are called, throughout the world. Ikaria is one such blue zone here in Greece. Some villages up in the mountains of Crete have also been studied extensively by the medical school here. And there are, of course, other blue zones in Japan, in Sardinia, Sicily, south of France, etc. So I think there is a common denominator among all these blue zones, and that's nutrition. That's way of living, lifestyle, specific lifestyle. Here in the Mediterranean basin, we have the Mediterranean diet that has been implicated in this exceptional longevity in those blue zones. Now, coincidentally, Mediterranean diet also includes a lot of fruits and vegetables that are rich in urolithin A, which is a mitophagy inducer, as I showed you.
Mm-hmm.
So one way of perhaps explaining, but I'm now brainstorming, and this is a speculation, is that diet is a very important factor because diet can affect such processes like mitophagy, autophagy in general, is induced by low calorie intake. So if we are under dietary restriction or caloric restriction, as they say, and this is something that is happening in these communities, in Ikaria, people are showing signs of dietary restriction, caloric restriction more specifically. Those effects actually can influence mitophagy, autophagy, and other detoxification processes in our cells. So they activate stress responses, oxidative stress responses, proteotoxic stress responses, etc. So by activating these responses, then you might survive for longer. So this might be an explanation why we have exceptional longevity in these blue zones.
Okay, thanks a lot.
Thank you, Professor Kavoulakis, for the question, and Professor Tavernarakis for the answer. And also, I can see a raised hand from Philippos Ververidis. Yes, please.
Yes, hello. First of all, congratulations, Professor Tavernarakis, not only for the talk, but for the excellent work. and I have actually two maybe oversimplified questions, but allow me to ask those because I had this curiosity since a long time, and hearing the ROS production in mitochondria made me rethink about this again. So, if I remember well, the Nobel laureate Pauli used to take 1 gram in the last years of his life, 1 gram of vitamin C. And I believe this was because he wanted to control the ROS. I don't know if it was well known at that time, and I don't remember how long he lived, but I think he lived quite long enough. And I wonder if you can comment on that. Okay, so if it is okay, lots of people even nowadays tend to consume. Since you mentioned about the dietary habits that we all have, antioxidants is one very important intake. That is one question, and the other one, perhaps I didn't hear it very well, but some connection I had with neurobiologists, I think they mentioned to me that we have neurons that they are replaced quite often. And if I remember well, you mentioned that neurons or some of neurons in the brain perhaps live for decades, maybe our whole life as humans. So which one is true? Okay.
Yeah. So with regard to the first question, actually, thanks for this because you give me the opportunity to clarify a few things about ROS and oxidative stress. There is additional complexity in these responses in that reactive oxygen species are also very important signaling molecules. So chelation, total absorption or blocking of reactive oxygen species is actually detrimental for cells. It's not good. So taking antioxidants, especially excessive concentrations or amounts of antioxidants is detrimental both for longevity but also for the overall health of an organism. And this is something that we have shown not just in C. elegans but also in other model systems. So it's not as simple as blocking or absorbing all reactive oxygen species, because if we do that, we essentially blind cells. We don't allow them to mount an antioxidant response. And this is a common mistake that people make and think when very simplistically we believe that taking antioxidants will protect us. Actually, excessive use or consumption of antioxidants can have the exact opposite effect. A little bit of ROS is actually good and prolongs lifespan. This is something that we have shown in multiple systems, not just us, but other labs throughout the world have shown that a little bit of oxidative stress can extend lifespan. So, for example, a little bit of paraquat can actually extend lifespan in C. elegans. Paraquat, as you know very well, is an inducer of oxidative stress and ROS production. So, it depends on, we have a biphasic curve. It depends on the dose. A slight dose will increase lifespan, will make things better, and will protect against neurodegeneration. But of course, if we overdo it, then we start getting detrimental effects. So the curve goes down. So oxidative stress, a little bit of oxidative stress is good. And completely suppressing oxidative stress responses by eliminating ROS, using, for example, antioxidants such as vitamin C, can be detrimental. And actually, Linus Pauling overdid it, and one of the reasons why his cancer was exacerbated, at least from what I understand, was this particular, or it is speculated that he consumed too much vitamin C. This is what I've read in literature and what I gather from what I've been reading. But perhaps perhaps you are familiar with a phenomenon called hormesis. Some of you might be. Hormesis is a phenomenon of a little bit of stress actually reinforcing stress responses. So a little bit of thermal stress can protect you against heat stroke, for example. And this is something that has been shown even in humans. It's acclimation. Same thing with people who climb to Mount Everest. They need to become acclimated first. So they need to be able to mount stress responses. So if we overdo it with antioxidants, we blind cells. We don't allow them to mount these responses, and this is bad. It's detrimental for cells. Now, regarding the so it's not as simple as taking an antioxidant and expecting to live longer or to battle neurodegeneration. It's not as simple as that. For each of us, the amount, the concentration of antioxidants that we should take in order to get an optimal effect might be completely different, and it's very difficult to really assess. You need to go through a very detailed biochemical analysis, detailed examination in order to determine what would be the optimal thing for each one of us. It's as if we are doing personalized, precision medicine approaches. Now, regarding the second question about neurons, indeed, in the nervous system, we do have some stem cell niches that will result in some limited renewal of some of our cells, especially in the olfactory bulb, in our nose. We have some capacity of neurodegeneration, but this is very limited, and it does not concern the majority of our nervous system. So, for example, hippocampal neurons cannot regenerate, and most of the central nervous system neurons cannot regenerate. Once a neuron is gone, it's gone. And if this neuron was involved in a specific memory, if this neuron dies, the memory dies with the neuron. And even if we somehow replace this neuron, that does not mean that we can recall the memory, because the engrams, the biochemical and cellular changes that encoded that memory trace in our brain are gone. Even if we could rebuild the brain, that would not mean that we would be the same person or we would have the same memories, the same experiences, etc. But neurogenesis is a very limited phenomenon in the central nervous system. It's really very limited, and it's an exception. It's a very small exception. There is some neurogenesis, indeed, yes, but it's a small exception. And most of our neurons actually accompany us throughout our lives. And it's indeed true that most of our neurons are born in the womb before we are born, and some of these neurons actually will die after we are dead. So we have the same cells with us for the duration of our lifespan. Let's say 85 years mean lifespan, or more than 100 years if some of us make it, if we are lucky.
If I comment something more, not a question, but actually this process of low-level stress is very important, not just for humans, but all kinds of organisms. And since I'm a plant scientist, let's say that we know that this low stress is also important for some processes, like maturation, for example, which is very important. Well, thank you very much. Thank you.
Thank you, Philippos, for the question, and Nektarios for the reply. And we have like 1 minute for a final question from Nikos Lidakis. Yes, please. A quick one, please.
Thank you very much for the excellent presentation. Professor Tavernarakis, first of all, if I remember correctly, Pauling had a very long life. He lived long after 90s, if I remember correctly. But anyhow, my professor had a very strong opinion about the discussion you had before, saying that probably Professor Pauling had very, very nutritious extractants. I mean, things that he didn't consume, just he consumed too much vitamin C, but how much of it remained in his body. Anyway, the question that I have is general also, and it is related to a prediction if you can make about how closer we get to fight this kind of diseases from your results and using results from other laboratories related to these issues around the world, if you can make any prediction.
Well, the more we understand these processes and the more we understand the fundamental reasons why these neurons degenerate, then I think the more close we are in developing a drug or a pharmaceutical that will be able to be used protectively, because neurodegeneration is non-reversible. If a neuron dies, it dies. There's no way of bringing back dead neurons. And as I mentioned earlier, neurogenesis is very, very limited and only concerns specific neuronal populations, not all neurons. But even if we somehow magically could bring back or regenerate lost neurons, that does not mean that we would bring back the information that was encoded in these neurons. So neurodegeneration is non-reversible. When a neuron dies, it dies for good. But we might be able at some point to act protectively. So, for example, if we are high risk for whatever reasons, perhaps family history, we have some inheritance reasons, our genes, the genes that we inherited from our parents, might make us more susceptible to Alzheimer's disease or Parkinson's disease, etc. Or if we believe that we are in a high risk group because we are exposed to environmental conditions that are contributing. For example, if we work in a polluted environment, pollution has been implicated in Alzheimer's disease. Malnutrition has also been implicated in Alzheimer's disease. So depending on this, we might be able to protectively get a drug that would upregulate these processes that are defending neurons, such as mitophagy, for example, in order to postpone the appearance of the phenotype of the disease or even block it altogether. But this is at a very early prognostic level. So it's similar to people who are high risk for suffering a heart attack. Then they take this aspirin pill that to some extent protects against such infractions. So similar strategy could be used for neurodegenerative diseases. But neurodegeneration, I should stress, is a irreversible process. So once it starts, the neurons that are dead are dead. So we need to act in a very early stage, at a very early stage. And this, of course, requires us to understand the reasons. Mitophagy is one reason, but I don't think it's the only one or even the major one. So we still need to study these processes. We still need to do a lot of fundamental research to identify all the mechanisms that are responsible for neurodegeneration. We have done considerable progress in recent years, especially. We have identified several processes, and now we need to develop a strategy that would perhaps influence in a combinatorial manner all these processes to allow cells to be fortified, to be more resistant to neurodegeneration. And as far as I know, certain pharmaceutical companies are actually doing exactly that. They are now trying to develop chemical compounds that target specific regulators of these proteins, of these processes, in order to be able to use them protectively against accumulation. So before the symptoms at early age, use these drugs to
Of
prevent
course, we have to
the
consider
disease.
side effects as well, huh?
Yes, of course. Yes, this is something that concerns clinical trials. Because administered in a non-cell specific manner, have adverse effects in other tissues that we don't want. So that's a big question.
Thank you very much.
Thank you very much, Professor Lidakis and Professor Tavernarakis, for the very insightful discussion. Unfortunately, we don't have time for more questions. And now I would like to give the floor to our rector of the Hellenic Mediterranean University to give us the final remarks. Thank you.
Okay, Nektarios, thank you a lot for this excellent talk. It was really very insightful, even for us coming from different scientific fields. It was really amazing hearing about all this research that you have done for facing these diseases that we have to struggle in the next years. So I have to admit that it has been really a privilege for me to know Professor Nektarios Tavernarakis over the last 10, 15 years. As you all know, he has been also the president of FORTH, the most prestigious research center in Greece, and one of the most prestigious researchers also in Europe. So, except of being a very distinguished scientist and researcher, Nektarios has been also a very successful, one of the most successful presidents of FORTH in the last decades. And he has combined his name also with the development and the success of FORTH. So, Nektarios, it has been a great honor to host you at HMU, at Athena Talks, and it was really a very excellent talk. Thank you a lot.
Thank you very much, Nico. It was a pleasure and an honor for me to be here. Thank you all for being here, and I hope it was not too tiring or too specific. I hope to see you all soon and have a face-to-face conversation.
Thank
Bye.
you.
Thank you a lot. Thank you a lot, Nektarios.
Have a nice
Thanks a lot.
weekend. Have a
Thanks
nice
a lot
weekend,
for
everyone.
the presentation and the discussion to everybody. And we renew our meeting for next week. And we have a speaker from the University of Oxford, Dr. Chrisanu Day, and he will discuss with us his research about light emitting diodes. Take care, and until next time, bye-bye.
Bye.
Bye.